Everyone. My name is Ashley, and I'll be walking through question 3 from the week 11 CSM worksheet, DNA sort. So basically, this question is asking us to run MSD and LSD radix sort on the following DNA sequences so the outputs sort in alphabetical order. So let's start with MSD sort. And before we get into the actual sorting process, let's talk about the basic algorithm behind MSD sort. So the logic behind MSD sort is that you're basically going through all the digits, or like letters in this case, from most significant to least significant in MSD, which in this case would be left to right, and sorting it at each place until the final thing is sorted in alphabetical order. It might not make that much sense now, but after I walk through this, hopefully it'll make a bit more sense. So let's start with the first iteration. So for the first iteration, we want to look at the most significant digit, which is the leftmost letter. So we know there's 4 possible letters in this case, A, C, G, and T. So we want to look for those in that order. So let's look for A's. So we find the first A is here. So we want to copy that out. And we find another A here. So we want to copy that out. If you find 2 digits of the same value, you want to tie break by having them stay in the same order as the previous list. Like for example, ACAG was ahead of ACAA in the list we're given, so we want to keep it so it's still ahead of ACAA in the next iteration. So that's all the A's. And I'm going to draw a line to signify that we're done looking at all the A's. And let's look for C's. So we find one C over here, the CTAG. And then we have one more C over here, which is CCTC. So that's 2 C's, and we found both of them. And so that's our C category. And then we are looking for G's. We find one G over here, which is GAGT. And that's all the G's. And then we look for T's, find one T over here, with TGAG. And that's all the DNA sequences after one iteration of this. And then for the next iteration, we want to look at the second most significant digit. In this case, we already sorted the most significant digit. So we know the most significant digit is already sorted. So that means we don't want to mess with it anymore, essentially. So we want to only sort within each category. So let's look at the first category, the A's, and then we see we're comparing a C and a C, and since they're both the same letter, we don't want to edit it at all. So we want to keep in the same order. So that's our A category, everyone's the same. Then let's look at the C's. So and for the second most significant digit, we're comparing C and T. C becomes is before T alphabetically, so we want to swap them. We want to put CCTC before CTAG. That's our C category, but since we sorted it, we want to split again. So now we know it's split in order again. And G and T only have one thing in each category, so we automatically know it's the optimally sorted version of itself. So that's what happens after we look through the second most significant digit place. So let's look through the third most significant digit place. So in this case, we have A and A again. And then, so we don't want to alter this. And every single other category only has 1. So we don't want to alter either. So it's the next version will be the exact same as this. I'm just going to copy and paste it to show that it has not changed. So that's what happens after the third iteration. And then we want to move to the fourth iteration, which is looking through the fourth most significant digits place, or essentially the least most significant digit place. So now we want to compare A and G. Thing is, A is ahead of G alphabetically, so we want to put the ACAA first before the ACAG. And then after that sorted, everything else we've already sorted into this correct category essentially. So it will remain in the exact same place. So I'm going to copy this down one more time. And then after this, we have our DNA sequences sorted in alphabetical order. So hopefully that helps you understand MSD sort. So let's move on to LSD sort. So LSD sort follows the same mechanism. We're still like looking at the digits place and then sorting, except we start with the least significant digit place instead of the most significant digit place and work backwards essentially. So let's walk through how to do LSD rate XOR on the same DNA sequence. So if we're looking at the least significant digit place, we want to compare the letters. So we want to look for A. So we find one A here. That's ACAA, so we want to put that at the top. Then we want to look for C's. We find one C, find CCTC. And then we want to look for G's. There's one G over here. There's another G over here. And there's one G over here. And after that, we find one T, and we want to put that at the end. So this is what a list looks like after you sort it through one iteration of least significant digit sort. You can see it's very different from what it looks like after one most significant digit sort in our previous iteration. One of the biggest differences is that like we're not sorting by category for least significant digits, because as they're going to be bouncing around a lot. Like for example, you see a C before A. Like in the final version, that cannot be possible. But if we split into categories, then C will always be before A, which is wrong. So we don't have any categories, so we're gonna have to look through an entire list every single time. So that being said, let's move on to the second least significant digit place, which is over here. So we want to look for A's. So we find 4 A's. And then so we want to keep them in order and move them to the top. And the same like tie-breaking scheme applies to least significant digit sort. If they're the same letter, you want to keep it in the same order that you find it. So here's all the A's. And then we want to move on. There's no C's, so we want to move on to G, which is G-A-G-T, and then the T, C-C-T-C. So that's what it looks like after one iteration of or like the second iteration, essentially. So this is what looks, and then, so let's move on to the third iteration. So we apply the same thing, but over to their least significant digits place. So we find one A, so we want to move that to the top. We find 2 C's or 3 C's. So first C is ACAA, and then ACAG, and then we find CCTC. And then we have a G, so we find TGAG, and then we find CTAG. So that's what it looks like after being sorted a third time. And now we're going to do the final iteration. So we do the same process. We look for our A's. We have our ACAA, ACAG, and then we look for our C's, CCTC, CTAG, and then we want to look for G's, GAGT, and then T's, TGAG. And then if you compare this to the previous answer, it's the exact same. So we're both sorted in alphabetical order. So that's how you perform LSD rate XOR and MSD rate XOR on the same list. And hopefully that helps you understand the sorting algorithm better.